Hello everyone. Today we will perform gene expression and regulation. We will use PhET gene expression essentials as the tool. Before we perform gene expression and regulation, we need to know about what is gene expression, what is gene regulation, what is affinity of binding, then positive transcription factor, negative transcription factor, and the tools that we require for performing the experiment. Yes, so gene expression means, as we know, the translation of DNA material into protein. For example, if we have a particular gene sequence that will be transcribed into mRNA first, then that mRNA, with the help of its codon, it will go for translation. So the journey from gene to protein is called gene expression. That means when the gene is expressed into protein, that's called gene expression. All right, now gene regulation. Gene regulation means regulating the expression of gene. It means controlling that which gene will be expressed now and which gene will not be expressed depending on the situation in the cell. So that is regulation. Regulation can be done by particular gene material or genetic sequences or different factors. Factors are usually proteins. They might be different chemicals. So affinity of binding means the affinity of binding of the chemicals to the genetic material or the transcription apparatus or the translation apparatus. Okay, because we all know that for transcription there are different materials and molecules which are involved in the whole process. So those molecules need to be bound with the transcription apparatus, right? For example, mRNA. In case of translation as well, different factors need to be bound with the translation apparatus. For example, ribosome, mRNA, etc. So this affinity of binding determines the rate of transcription and translation in cells. Okay. Now, what is positive transcription factor and what is negative transcription factor? Positive transcription factors are factors that when involved in the transcription process, it actually helps the transcription process to forward. And negative transcription factor is actually a regulatory factor which will stop the transcription or decrease the rate of the transcription. All right. So what we need for the experiment, we need the device, we need Microsoft Edge. You can also use like Google Chrome or Firefox. That will be useful. PhET gene expression essential, usually we used to download and install it before, but now it is an open source material, so we do not need to download that. Sniping tool is for taking the screenshot. So for PhET gene expression essentials, we have a link provided at the end of the material. This one. Okay. Copying the link. Yes. So after I have copied the link and I opened a new tab and I have pasted the link here, and this is PhET gene expression essentials. Now getting back to the manual. Yes, so according to the methodology, we need to download the PhET gene expression essentials, which is not a requirement now because it's an open tool now. So number 2 is go to multiple cells. So this is the homepage image that you can see, and now we will go to multiple cells and check the average protein level versus time. Okay, going to multiple cells, check the average protein level. This is the average protein level with time. So we can see that from the cells, the proteins are being produced and the level of protein is demonstrated here in that graph. Okay. So this is the average protein level versus time by this organism. Okay. This is a bacterial cell. Now the question is, what do you see when you increase the mRNA destroyer level and protein degradation level in case of single cell? So you need to click on the bars here. So they will be opened. Now they asked in the question that what do we see in case we increase the mRNA destroyer level and we increase the protein degradation level. So with time, the protein that is produced by this bacterial cell will be degraded. So when the degradation rate of the protein is high, then you see this is the level of protein with time. So when I decrease the degradation level of protein, it is increasing a bit. So what you need to do is to see the protein level versus time when you increase the mRNA destroyer and when you increase the protein degradation rate. Take the screenshot of the process or just the graph, paste it to your manual, and discuss what you have seen. For example, I have seen that the protein, when I have increased the mRNA destroyer, the protein level in this graph is going down. right? And also when the degradation is fast, it's again going down. All right, now what do you see when you increase the affinities of RNA polymerase and positive transcription factor in case of both single and multiple cells? So what do we see when we increase the polymerase and positive transcription factor? So this is the polymerase binding. If we increase the polymerase and we also increase the if we increase the polymerase, this is polymerase, and if we increase the positive transcription factor, then I'm making it high. Then of course the protein production, average protein production by this bacterial cell will rise with time, because the positive transcription factor, as we have mentioned before, that the positive transcription factor will increase the transcription process, will increase the rate of the transcription process. And for polymerase, we also know that it is the enzyme which is involved in the transcription process, which initiate the transcription process. So this is for the, when I make it high, both the positive transcription factor and the polymerase, then this is the graph. The protein level is getting high. So this is in case of single cell. You need to take a screenshot of this process, then make it for multiple cell, right? Because in the question we need to find it out for multiple cells as well. So for multiple cell, this is the cell level. I will increase the cell. See, it's getting multiple. In case of single cell, we had this result. Can you see that the graph is a bit scattered, right? It's fluctuating. In case of more cells, when we try to see the protein production by more cells, many cells, then the protein level is high, but the graph is a bit smooth, right? It's not that much scattered. It's a bit stable, because when we take the count for multiple cells, we get an average protein level which is more stable. The fluctuations in the protein production is less visible then. So you need to take a screenshot here and add to your manual and discuss what you have observed, okay? Now, what difference you see between single and multiple cell response? I think the best difference that we have observed that the single cell response is very fluctuating in terms of the change in the environment, change in the transcription factor, and mRNA destroyer, so many other things. In case of multiple cell, the graph was smooth. The response of the cells in terms of protein production was stable. Going to the next level, go to mRNA. All right, I'm going to mRNA. Yes. Yeah, so this is the page for mRNA. Concentration and affinity of positive transcription factor are high and affinity of RNA polymerase is low. So we need to fix the affinity of PTF, that is positive transcription factor, high, and affinity of RNA polymerase is low. So we need to fix these things and then attach the screenshot here. That is for you to do. I'm not attaching any screenshot. So transcription factor is high, but RNA polymerase is low. So let's fix the RNA polymerase to low and positive transcription factor high. So these yellow things, these are positive transcription factor. And you can see these are RNA polymerase, these blue balloons like things, these are RNA polymerase. So when RNA polymerase is very low and positive transcription is high, positive transcription factor is very high, the RNA polymerase is not binding to the mRNA. As we know, for binding to the mRNA, for transcription, we need the RNA polymerase to bind to the mRNA first. But when the affinity of RNA polymerase to the mRNA is very low, then they are not being bound to mRNA and the transcription is not being started, right? So you need to take a screenshot of this phenomena and you have to add it to your manual. Now, concentration of positive transcription factor is high, affinity of positive transcription factor is low, and affinity of RNA polymerase is high. Concentration of positive transcription factor, we need to fix it in high. Concentration of positive transcription factor. So this is the concentration of positive transcription factor, which is high. I've already set it high. Then affinity of positive transcription factor is low. So I'm fixing it to low. Then affinity of RNA polymerase is high. Okay, it's high. Now let's see what happens. So you see the positive transcription, because the affinity of positive transcription factor is low. It's not being attached to the mRNA. Though the concentration is very high, even if there are lots of positive transcription factor, but they are not being bound to RNA polymerase and the mRNA because the affinity is low. So affinity is a big factor. Even if we have enough transcription factor, they will not work if the affinity is not working between them. And then you see the RNA polymerase is also in very high amount here. The affinity of RNA polymerase is also high, but they are not being bound to mRNA. So this is the mRNA. Okay. Now, so you will take the screenshot of the process and add it to your manual. And now, concentration of positive transcription factor is high. Okay, I think this is the, okay. So in number C, we need to fix in the concentration of positive transcription factor as high. So the concentration of positive transcription factor is already high. Okay. Then affinity of positive transcription factor is low. Affinity of positive transcription factor is already low, yes. Then transcription factors, the concentration and affinity of negative transcription factor are high, and affinity of RNA polymerase is high. So negative transcription factor is here. I'm clicking on this. So an extra bar is here. This extra box is containing the details for the parameters for negative transcription factor. So according to the manual, we need to fix the negative transcription factor concentration and affinity as high. So the concentration of negative transcription factor is high, and affinity of negative transcription is already high. And RNA polymerase binding affinity is high. See, even if the RNA polymerase is in abundance and also the positive transcription factor is high in number, but when the affinity for positive transcription factor is low and the affinity of negative transcription factor is very high, as you can see, there will be no transcription. So that's the result. You can take a screenshot and add it to your manual. Now, D, concentration of positive transcription factor is high. All right, concentration of positive transcription factor is high. Affinity of positive transcription factor is also high. So I'm making affinity of positive transcription factor as high so that it can bind to the mRNA. Then concentration and affinity of negative transcription factor are high. So negative transcription factor concentration and affinity both are high. And affinity of RNA polymerase is also high. So affinity of RNA polymerase is also high. Still no transcription is occurring here, as we can see. The affinity of negative transcription factor is high, the red ones. So it is already bound to the mRNA. The affinity of positive transcription factor is also highest. And concentration is also highest, but it is not being bound to mRNA because of the negative transcription factor. That means if there is enough negative transcription factor in high concentration and with high affinity, that means it will not allow the positive transcription factor, the yellow ones, to bind to the mRNA. And also it is not letting the RNA polymerase to bind to the mRNA. So you can have a screenshot and add it to your manual. Then Then number 4, go to expression. Okay. You see, when I have lower the concentration and affinity of negative transcription factor, so there is no negative transcription factor, and the positive transcription factor concentration and affinity binding is high, then RNA polymerase affinity binding is also high. You can see that the mRNA is being produced from the DNA. Okay. Now, getting back to the manual, go to expression. So I'm going to expression. Now, so this is the DNA. It's the regulatory region of the DNA. And this is the region that will be transcribed to mRNA. Okay. So the whole portion covering the regulatory region and the transcribed region is being called the gene. So you can see there is a toolbox having the positive transcription factor, which will help the transcription. There are RNA polymerase that will also help the transcription, and that will actually initiate the transcription. There are ribosomes, which is another transcription apparatus, which is actually translation apparatus, which we will require for translation. Then mRNA destroyer. These are the factors that destroy the mRNA and decrease the protein production by destroying the mRNA and decreasing the mRNA level. Because we know that mRNA, ribosome, these are the requirement for protein translation. Negative transcription factor is a factor when we have already seen in the mRNA section that when it was bound to the DNA, it resists the binding of positive transcription factor to the DNA, and no transcription has occurred, right? This toolbox, this is not the toolbox, this is actually the protein box, where we can see there are 3 types of protein of 3 different shapes. These are 3 protein types. We have 3 different genes actually. That's why we have 3 protein section. So I'm getting back to the first gene. Collect as many protein as possible for 3 protein types within 15 minutes. No residual mRNA, RNA polymerase, positive transcription factor should be present. These are the conditions. Negative transcription factor should be placed on the regulatory region of the DNA straight after you are done. Then provide the screenshot of your page after completion of protein collection. Okay. So what we need to do is we need to start producing the protein. So first of all, we need to take out from the toolbox the positive transcription factor. Okay, so it's bound to it. Now what do we need? We need the RNA polymerase, right? RNA polymerase. Yes, so you can see the RNA polymerase has produced the mRNA, which is single-stranded. Okay. Now I'm placing it back. So this is my mRNA. To produce the protein, I need what? I need the ribosome. I need to attach it to the mRNA, and it will produce a beautiful protein. This is the protein, and I will put it in according to the shape in the protein box. I will put it there. So yeah, this is the way I need to make it. Now, so this is the protein production for the first gene, gene 1. After you are done, you need to destroy the mRNA. Place the thing back. Place the ribosome back. Place the regulatory, the positive transcription factor back. Yes, moving to the next gene. So this is gene 2, right? Now we have 2 positive transcription factor here. I will place it there. We have RNA polymerase, which will produce mRNA, which will read the DNA and produce mRNA according to the DNA sequence. Then I'm getting I'm placing the RNA polymerase back here, and I will add the ribosome to the mRNA. See, so this is the second type of protein. So I will place it in the protein box. I will place the ribosome back, and I will destroy the mRNA. Yes, placing the mRNA destroyer back. So as you can remember that we are supposed to place the negative transcription factor after we are done with the production. I have to do it for the previous gene as well. Negative transcription factor, placing it here. Moving to the next gene. This is gene 3. Okay, so for gene 3, I need positive transcription factor again. That will help the binding of RNA polymerase to the DNA. Before that, I need to, it's not being bound because I have to place another positive transcription factor here. Yes, now RNA polymerase. See, the mRNA is ready. I'm getting I'm putting back here. Now I will have the ribosome to the mRNA, and as you can see, protein is already produced. So I will place the protein to the collection box, and I will destroy the mRNA and placing the ribosome back, placing the mRNA destroyer back to the toolbox. Because I'm done, I will place the negative transcription factor on the DNA. Okay. Now you can see I have all types of protein in the protein collection box. So what you need to do is within 15 minutes, collect as many possible as you can, and take a screenshot with time, with the starting time and the finishing time. Okay. Yeah, I hope you will enjoy producing protein by this tool. All right. Thank you very much.